So our next speaker is Skulas-Ray, and she is an assistant professor in the School of Nutritional Sciences and Wellness in the College of Agricultural and Life Sciences.
Thank you. Okay. Everyone's still with us. Great. So I'll be speaking about resolution of inflammation. And to start, I think we can all appreciate that chronic diseases in this country are a huge problem, very expensive. The typical person has at least one chronic illness. Some of us have more than one. And chronic inflammation is at the heart of many of these so-called non-communicable diseases. So I'm going to talk a little bit about inflammation in acute and chronic disease pathology, what I mean by resolution of inflammation, and how that informs wellness. So inflammation as a term refers both to this normal protective response that we have, but also the pathological response. And we think of it as these pillars of heat, redness, swelling, pain, loss of function. And what's happening is we have a stimulus, whether it's a pathogen or an injury, and changes in gene expression that lead to a whole programmed inflammatory response that can be throughout the entire body or localized, as we see here. So we can get to acute inflammation in many different ways. This is my list of things that we have to tell research participants not to do prior to coming into testing visits, because we've learned if we don't specify to not do this, we will have people do 50-mile bike races the day before a testing session. But what we really care about is this chronic inflammation that doesn't resolve. This is where we get increases in risk of cardiovascular disease and many of these sort of pathological changes that accumulate with biological aging. And we can measure this with this marker C-reactive protein. It serves as a really nice way to get at the systemic levels of inflammation throughout the entire body. And it's not just the inflammation, but it's how it drives all these different pathological processes that we care about that have implications, again, for cognitive decline, cardiovascular disease, cancer, all these very expensive, incurable conditions of adverse aging. So the other side of the coin is resolution of inflammation, and we now have sort of a sense of pillars of what that means. So in contrast to inflammation being the sort of, process that leads to your pain, loss of function, resolution is a programmed removal of dead tissue, immune cells that are no longer needed, and it's restoring that homeostasis. It's bringing us back to where we want to be. So we want to have that protective immune response, but then we want to switch over to this resolution process. And it's a tightly coordinated event where we switch from initiation to resolution. And it's driven by these specific lipid mediators, lipoxins, resolvins, protectins, and mucins. The important thing to know is that these are derived from 20 and 22 carbon fatty acids from our diet. Because we're humans, we can't put in these double bonds for these fatty acids, so we're dependent on consuming them. If we don't eat those omega-3 fatty acids, we cannot make those compounds circled in blue. And that's very important because this is a tightly regulated response, and those compounds are important for driving it. So this is a resource called the Atlas of Inflammation Resolution, where all of the knowledge that's been generated on the coordination of this process has been sort of put together. So if you're curious, this is a really nice resource. And we know a lot about these compounds and how they act. What you might notice from this list is these are all animal models. But we need these to really understand the important role that the resolvins, lipoxin, protectins, mucins play in all these different disease states that we care about, all these chronic inflammatory diseases. I didn't know all the other talks would have E. coli, and I bet you didn't know mine would either, but the reason why we use animal models so much is because we can induce inflammation in a very controlled way. And so what we did in our research was we translated that to humans, and we used a purified endotoxin that was derived from E. coli at a very low dose that was injected intravenously to induce inflammation throughout the entire human. And that way we could look at the inflammatory initiation and that resolution phase. And importantly for us, we wanted to see if we could modulate it. We wanted to change that response in a beneficial way. So we gave omega-3 fatty acids to people who didn't eat a lot of oily fish, which is actually very common in the United States. Most of us don't eat enough fish. So we took low fish consumers, we gave them a very high dose of omega-3s. They were healthy adults, and it was a randomized crossover with a placebo control. So they got endotoxin injection twice. Everybody came back for the second injection. And the hypothesis we had going into this was that because omega-3 fatty acids are the precursors for all these important compounds, we were going to suppress that inflammatory response. What we found was absolutely no suppression. So the blue line is after the omega-3s, the red is after placebo. And these are 20 people who were exposed to endotoxin twice, with and without that fish oil. And they didn't know and we didn't know what they got. But we had the exact same acute inflammatory response measured by CRP and by cytokines. So, I think the first reaction was, oh no, it didn't work. But what we did get was activation of these pathways. And while we can't really measure the resolvins and protectins well in the blood, we can measure those compounds that are circled in red. So we're calling them pathway markers, and they're a really good measure of what's happening with these pathways and the resolution of inflammation. And what we found was that 18-HEAP, which was made out of EPA, was increased just as a result of giving that high dose of fish oil, but then it further increased during that inflammatory response. This is really important because it illustrates that we were getting activation of resolution processes in the body. And 18-HEAP is bioactive in and of its own right. So in mouse models of induced heart attack, it prevents fibrotic remodeling, which is really important because a lot of times people have sort of scarring after heart attack and then arrhythmia later. This compound prevents that sort of adverse scar formation. So the important thing in talking about wellness versus health and sort of these different paradigms, pro-resolving is not immunosuppressive. It's not even technically anti-inflammatory. It's about optimizing function and resilience. And we've had a lot of discussions about what exactly is wellness. And I think we can consider it similar as sort of an optimized function and resilience. And we talk about resilience a lot, I think, in these different thematic areas, because what we want is not to suppress the immune system. We want it to work very well, but we also want our tissues to return to their sort of healthy, normal homeostatic state. And this idea of promoting resilience is very different than disease prevention. So it's not disease treatment, it's not even disease prevention. It's sort of this idea of promoting resilience. This was a figure I made 8 years ago to think about this, right? So I debated whether or not to put my slide of, for every complex question, there's an answer that is simple, clear, and wrong. So we always want to change this inflammatory response. And so omega-3s are what I showed you, and that's a very small piece. But we're also interested in these other determinants. So what you're looking at here is pretty much all the One Health ideas of, we have our sort of environmental exposures. We have also our stress levels, which are influenced by our companion animals. But all of these factors are going to influence our inflammatory state, our immune function. And we're going to have, the microbiome effects and also our individual genetic variability. So as it relates to the wellness and One Health links, we've been doing different collaborative projects around the themes of sustainable food systems, innovation in production and processing. An example of that is these grass-fed beef project that we have, where we have these cattle that are actually very well suited for our very dry climate, and they are actually helpful for preventing fires, and they can be finished out on the range. And they're healthy, they protect their calves, and they're really just, I think they're a good One Health example from the environment, the animal welfare, and the human health, because by finishing these cattle on range and grass, not with hay, what we end up with is higher concentrations of omega-3 fatty acids in their tissues. And so when we eat these grass-finished beef, we will be ingesting omega-3s. So it's all connected. We're also very interested in innovation in the built environment for wellness, and to that aim, we're sort of building up this collaboration with my partner in the community who's a lipidologist. And we're calling it the Partnership for Optimizing Wellness and Empowering Research, just to have a cute acronym of POWER. But we'd like to build on these themes and understanding them better. And in addition to wellness being this idea of both performance and resilience, we can also think of it as the process or path owned by the individual, according to their values, their choices, and their priorities. So in that way, it becomes very personalized. And I think the distinction is nutrition, exercise, and sleep are owned by the individual. It's something that they take home with them. They do in their own lives. It's their choice on, based on their resources, their knowledge, their support, to implement these in their activities. Okay, and so these are related to resolution of inflammation. I like Q&A a lot, so I want to make sure I get to that. How many minutes do I have? We're good? We're good. Okay, good. So I'm ready for questions.
Hi, thanks. That was really interesting. My name is Melissa Furlong. I'm in the College of Public Health. So I think in the beginning you talked a little bit about chronic inflammation and acute inflammation. And in your study, it looked like you were measuring the differences in acute inflammation after your omega supplements. So I know, like, for instance, physical activity induces an acute inflammatory response, but ultimately results in lower inflammation over time. And I just wanted to ask if you had looked at differences in chronic inflammation after long periods of time after the challenge.
Okay, very nuanced question with a lot of components. One thing I'll point out, so the idea of the chronic inflammation versus acute, we went out to 168 hours, 1 week post-injection to follow that entire inflammatory response curve. But what I can point out is all the symptoms were confined to 2 hours when that cytokine peak occurred, and there was a lag with the CRP. And these were healthy people, so they didn't experience any chronic inflammation. So that's still a challenge, and one of the conversations we've had around that is if we can use a more sustained stressor. So I didn't really go into some of the other collaborations, but with the Center on Aging, we were trying to look at whether frailty can be predicted by these compounds, and above and beyond that, responses to surgical stress, because that would be a more sustained physical stressor. But we'd also like to look at vaccination and some of these other sort of more extended models. The benefit of this endotoxin challenge is how precise and control that we could get this. Whereas, if we do a vaccination, the second time someone's vaccinated, they won't have the same response. But it'd be really nice to do this with this sort of research with shingles vaccine, because that is a 2-dose and the symptoms are just so strong and we get a robust signal. So there's always the challenge of finding the signal to noise ratio that works for human models. But yeah, very good question. And in Europe, they've done repeated exposures to try to get more of a sustained inflammatory response. But the United States FDA does not like that. So we're a little bit limited to healthy people and this very acute exposure. Did I get your question? Yeah, and that's important. Exercise also induces productions of resolvins and protectins, myrisins, all these compounds. So looking at the influence of exercise, adequate sleep. Actually, in our pilot study, I was hoping we would see this more broadly, but we looked at saline versus LPS, and I noticed that some people had lower responses, and I thought it was going to be more easily modeled by physical activity or age. But in talking to these people, I realized The ones who had the highest inflammatory responses reported having young children at home. And for them, being injected with endotoxin in our facility was sort of a spa day, because they were allowed to sleep and we brought them food and we fluffed their pillow. So I think that they came into it more sleep deprived, and that was sort of reflected. And we would expect to see too that, oh, that was one of my favorite slides about misinformation. So yeah, I'll just leave that one up there. We get a lot of, I mean, it's in nutrition, we get a lot of opportunities to practice talking to the public. And so this is something that we have to address a lot, is the whiplash people get about nutrition information. But the advisory is actually mine. But that was, when I updated, that was the most recent one. I thought that was a good attention grabber. Thank you.
Thanks so much.